This video explores how the curvature of cell membranes influences the distribution of cholesterol and other lipids in the membrane leaflets during processes such as vesicle fusion and protein embedding. Understanding the central question is how the shape of the membrane at fusion pores, or protein-induced deformations, influences the spatial distribution of cholesterol in the bilayer leaflets. This matters because cholesterol plays key roles in modulating membrane properties and protein function, and its localization can regulate membrane curvature and fusion events. The authors approach this by decomposing the total membrane curvature into two principal components, one due to bending radially from the pore axis and another from bending around the axis of symmetry. These curvatures affect lipid packing preferences differently because lipids vary in head group and tail area. Through theoretical analysis, they examine how these curvatures compete to favor or disfavor cholesterol in specific membrane regions. The focus is on the cytoplasmic leaflet, where the two principal curvatures have comparable magnitude but opposite signs. Creating a complex pattern of lipid distribution, the approach helps to clarify previously conflicting findings about cholesterol localization near membrane fusion pores. Figure 2 illustrates the two principal curvatures of the cytoplasmic leaflet, labeled CIR and CIQR, as well as their sum, the total curvature. The first curvature, CIR, is positive at all distances from the pore axis, reflecting bending in the radial direction, while the second curvature, CIQ, is negative at all distances, representing bending around the axis of symmetry. Because these curvatures have opposite signs but similar magnitudes, their sum, the total curvature, changes sign depending on the distance from the pore. This interplay means that the membrane shape produces competing effects on cholesterol distribution. The positive CIR curvature favors lipids with large head groups and small tails, which tends to reduce cholesterol content, since cholesterol increases tail area. Conversely, the negative CIQ curvature favors lipids with small head groups and large tails, thus favoring cholesterol enrichment. The total curvature profile, Figure 3, presents the theoretical results for the mole fraction of cholesterol in the cytoplasmic leaflet as a function of distance from the pore axis. The horizontal dotted line indicates the cholesterol mole fraction in a flat membrane, set at 0.30. We observe that the cholesterol mole fraction near the pore axis initially shows some variation, but overall the net change compared to the flat membrane is negative across the cytoplasmic leaflet. This result builds on the earlier analysis of the competing principal curvatures and confirms that the total curvature effect leads to a reduction in cholesterol content within this leaflet near the pore. This theoretical finding provides insight into how membrane shape modulates lipid organization during fusion events or protein-induced membrane curvature. In Figure 4, the authors extend their theoretical analysis to other lipid components in the cytoplasmic leaflet, presenting mole fractions for phosphatidylethanolamine, PE, cholesterol, phosphatidylserine, PS, and phosphatidylcholine, PC. The results are plotted with horizontal dotted lines indicating their composition in a flat membrane, compared to cholesterol, which shows a net decrease in mole fraction consistent with Figure 3. Other lipids respond differently to membrane curvature. This multi-parameter lipid distribution highlights the complex interplay of membrane shape and lipid preferences. The authors note that although their curvature-based model captures these effects, some observed behaviors, like simultaneous decreases of cholesterol in both leaflets, seen in simulations, remain unexplained. This may be due to assumptions such as constant membrane thickness in their theory versus bilayer thinning near strongly curved regions. Furthermore, direct protein-lipid interactions, such as with the piezo-1 protein embedding, are not included but may also affect lipid distribution. Overall, Figure 4 enriches understanding of lipid rearrangements induced by curvature in the cytoplasmic leaflet. Figure 5 shifts focus to the exoplasmic leaflet, showing its two principal curvatures, COQR, and their sum, the total curvature. Unlike the cytoplasmic leaflet, here, the curvature from bending around the axis of symmetry, which is negative and proportional to the inverse distance from the pore axis, is diminished because the exoplasmic leaflet is located further from the symmetry axis. This negative contribution never exceeds that of the radial curvature, which remains positive and dominates. Consequently, the total curvature in the exoplasmic leaflet remains positive at all distances. This positive total curvature favors lipids with large head groups and small tail areas less compatible with cholesterol, suggesting that cholesterol distribution will differ from the cytoplasmic leaflet. The authors interpret these curvature properties as underlying the lipid composition changes predicted in the exoplasmic leaflet during Figure 6 illustrates the mole fractions of cholesterol, sphingomyelin, SM, and phosphatidylcholine, PC, in the exoplasmic leaflet as functions of distance from the pore axis. Each component is compared against their respective baseline mole fractions in a flat membrane, indicated by horizontal dotted lines. The results reveal that changes in lipid mole fractions are more pronounced in the exoplasmic leaflet than in the cytoplasmic leaflet, consistent with the curvature profile shown in Figure 5. Specifically, cholesterol mole fraction increases near the pore axis in the exoplasmic leaflet, reflecting the positive total curvature that favors its enrichment. This behavior aligns with previous observations in membranes with embedded proteins like piezo-1. Comparing Figure 6 with Figure 4 highlights an important distinction. Cholesterol decreases in the cytoplasmic leaflet, but increases in the exoplasmic leaflet. The authors point out that while their theory captures these curvature-driven trends, discrepancies with simulations arise due to bilayer thinning and protein interactions not included in their model. Nonetheless, these results clarify the mechanistic link between membrane shape. In summary, this theoretical study elucidates how membrane curvature modulates the spatial distribution of cholesterol and other lipids within the cytoplasmic and exoplasmic leaflets of bilayers undergoing complex deformations, such as fusion pore formation or protein embedding. The analysis decomposes membrane shape into two principal curvatures whose opposing effects compete in the cytoplasmic leaflet, ultimately predicting a net cholesterol decrease there. Conversely, the exoplasmic leaflet exhibits a positive total curvature that favors increased cholesterol content near the pore axis. These curvature-driven lipid rearrangements provide new mechanistic insight into observed lipid heterogeneities during fusion and mechanosensing processes. The work also highlights limitations of curvature-only models, including the need to consider bilayer thinning and direct protein-lipid interactions, which can alter cholesterol localization. Nevertheless, the theoretical framework presented offers a valuable foundation for understanding how membrane geometry regulates lipid composition, with implications for membrane fusion, signaling, and protein function in cellular contexts.